Good morning, everyone. Today topic biomolecules. In biomolecule, we discuss about important biomolecules, that is carbohydrates. So in this today video lecture, we will discuss only monosaccharide carbohydrate. First, what is carbohydrates? Generally, in name only, it is suggest hydrates means water. Hydrates of carbon is called carbohydrate. Here I am writing carbohydrates are nothing but hydrates of carbon. Means like this. Hydrates means water of carbon is known as a carbohydrates, or simply we can write like this: carbon and it should has hydrogen 2n and oxygen n. So in carbohydrate 1 is to 2 is to 1 must and should be present. The ratio of carbon, hydrogen, oxygen must be 1 is to 2 is to 1. Then only it's said to be as a carbohydrate. But there is some exception is there. But there are some exception. There are some non-carbohydrate molecules, but also they contain 1 is to 2 is to 1, but they do not consider as a carbohydrate. For example, here acetic acid, acetic acid, acetic acid. So here acetic acid formula is CH3. So here, if you observe this, in which carbon, hydrogen, oxygen ratios. So here, in which carbon to hydrogen, here 3 and here 4. So hydrogen 4 and oxygen is 2. So 1 here 2, here 1. So 1 is to 2 is to 1. But still it doesn't consider as a carbohydrate. And one more exception of non-carbohydrate molecule, they also contain 1 is to 2 is to 1 carbon, hydrogen, oxygen ratios, that is lactic acids. Lactic acids. Lactic acids. So formula here, lactic acid C3. So like that 3, 1 is to 2 is to 1. Still it is not a carbohydrate. So these two are most important exception of carbohydrate, and they contain 1 is to 2 is to 1 ratio. But must and should every carbohydrate molecules contain 1 is to 2 is to 1 carbon, hydrogen, oxygen ratios. And coming to here, the carbohydrates also known as sugar. They also known as the sugar. And here these carbohydrates are definition for important definition for carbohydrate is they are polyhydroxyaldehyde means like CHO, or they are polyhydroxy means like this C double one O ketones. So this is the definition for carbohydrate, polyhydroxyaldehyde, or they are polyhydroxy ketone. For example, here most important concept, hydroxy. So OH. Note here, hydroxy group must and should be more than 2. So here hydroxy group, note hydroxy group must be poly, means more than 2. Hydroxy group must be more than 2. More than 2. Then only it's said to be as a carbohydrate. Generally in carbohydrates, there must be either aldehyde group or ketone group should be present. So in carbohydrate, there must be either CHO means aldehyde group or ketone group. Ketone group must and should be present in carbo. And remember one important note here. So every carbohydrate end with a suffix with OSC. Then it to be carbohydrate. So for example, glucose, OSC, carbohydrate. For example, fructose, OSC, carbohydrate. For example, sucrose. So like that. So every end of this molecule, if they have OSC, then it's said to be as a carbohydrate. And one more important note, if the suffix end with A, it must and should be enzymes. For example, sucrase, ASC, means it is an enzyme which hydrolyze the sucrose, like that. And for example, fructose, or invertase, acid, and enzymes. So maltase, or lac. So every end of this or suffix of this enzyme end with AAC, then it's said to be as a enzymes of carbohydrates. Or end with OAC, then it's said to be as a molecule of carbohydrates. Okay, right. So now we move on classification of enzymes. Carbohydrate. Classification of carbohydrates. Generally, carbohydrates classified into three important parts. First one, monosaccharide, mono, and second one, oligosaccharide, oligo. And third one is most important, poly. So in this video lecture, we only focus on mono. So here, monosaccharide means which cannot be further hydrolyzed. These monosaccharides cannot be further hydrolyzed. Hence, they simply said to be as a sugar. They are simple sugar. And coming to disaccharides, which can further hydrolyze. This disaccharide can further hydrolyze into monosaccharides. They can further hydrolyze into monosaccharides. And these oligosaccharides are made up of or comprises more than 2 to 10 monosaccharides. They contain or comprises 2 to 10 monosaccharides units. Now coming to polysaccharides. So polysaccharides can be further hydrolyzed. They also can further hydrolyze. They can further hydrolyze into either oligosaccharide, oligo, and monosaccharide. And these polysaccharides, which can made up of or contain more than 10 monosaccharides, which made up of or comprises more than 10 monosaccharides units. Monosaccharides units. So likewise, we have different types of carbohydrates based on their hydrolyze further. Yeah. So here we will focus only on in today's lecture we focus on only monosaccharide. So now we'll discuss about monosaccharide. Monosaccharide. Monosaccharides are simplest form of sugar. They are simplest form of sugar, and it cannot be further hydrolyzed. It cannot be further hydrolyzed. So here we have various types or examples of monosaccharides. Here, examples of monosaccharides. In that first we will discuss about. So if monosaccharides which is contained with 3 carbon, they are called triose sugar, or simply we can use trioses. And this carbohydrate contain carbon number 3. So here we should apply a formula like Cn and O 1 is to 2 is to 1. So like that, if we can frame this triose sugar like carbon number 3, then hydrogen must be 6 and oxygen is 3. C3H triose sugar contain the formula C3H6O3. And triose sugar is among the monosaccharide. Triose sugar is the simplest sugar. And these triose sugar, so just now in definition we discuss in carbohydrate are either aldehyde group or ketone group must be present. So based on which triose sugar are classified into two important categories like this. So in triose we have aldose form of carbohydrate and ketose form of carbohydrate. Aldose form and ketose form of carbohydrate. So here ketose means COO. Here aldose form is CH. For example, the aldose form of triose sugar is glyceraldehyde. Glyceraldehyde. So glyceraldehyde is a aldose form of triose sugar. Now coming to ketose form of triose sugar is dihydroxyacetone. Dihydroxyacetone. So dihydroxyacetone is a ketose form and glyceraldehyde is a aldose form of triose sugar. For example, if we write their chemical formula means like this. So first carbon always in aldose CHO. Here I am writing aldose form glyceraldehyde formula C carbon and here CH and C. And coming to here dihydroxyacetone phosphate has. So if we interchange means here at second carbon OH if it is present above the then like this. Sorry, like this and here C at second carbon we have ketose group. In here in aldose form at first carbon we have aldehyde functional group, but in aldose ketose form the functional group like keto present at the second carbon, and then CH2. So this is the dihydroxyacetone phosphate formula, and here glyceraldehyde. Glyceraldehyde, this one glyceraldehyde, and this one dihydroxy acetone, dihydroxy. Okay. So likewise we have two forms of triose sugar. Now we move on another example for monosaccharide. So that is tetrose sugar. Second example, tetrose sugar. So tetrose means carbon number 4, or simply we can use tetrose. So if it contains carbon number 4. So if you apply the formula like carbon number 4 and hydrogen 8, oxygen 4, the carbon number, and a formula for tetrose sugar. And in tetrose sugar also we have two forms, like here in which tetrose sugar, here I am writing, yeah. So aldose form of tetrose and ketose form of tetrose, C double bond O, here C, yeah. So aldose form of tetrose sugar, best example is erythrose, and here ketose form of tetrose sugar is erythrulose. Okay. So if you draw the formula for erythrose like this, so always in aldose form CHO is functional group present at first carbon like this, CHO, and simply we can write CHOH and CH and CH2. So this is the formula for erythrose. So if you write the erythrulose formula, simply we can change that carbon number 2 we have functional group, that is ketone group. So CH2, C double bond O, and as it is CHOH and CH2. So this is all about the erythrulose, and this one erythrose. So likewise erythrose and erythrulose both are tetrose sugar. Now we coming to one more third example, that is pentose sugar, means they contain 5 carbon compound. Pentose sugar. So this pentose sugar or pentose, simply we can use pentose, and they contain carbon number 5. If you apply the formula, then carbon number 5 means hydrogen double 2n, means 10, oxygen is 5, C5H10O5. It is a pentose sugar formula. And in pentose sugar also we have two forms. So like in pentose sugar we have two forms, yeah. So example there, aldose form of pentose sugar, means CHO group, and ketose form, ketose form means C double one O. So aldose form of pentose sugar, best example is ribose sugar, very important, ribose sugar, ribose. Now coming to ketose form of pentose sugar is ribulose. And here see every aldose and ketose form both are every like in uh, triose or tetrose or pentose or hexose, aldose form and ketose form both are isomers. So both are isomers to each other. Remember, isomers. So here also this tribose and ribulose both are aldose form or ketose form are isomers only. Okay, fine. So generally this ribose sugar are seen in RNA, ribonucleic acid, RNA, non-genetic matter. In RNA we have seen this ribose sugar, very important. And these ribulose are generally seen in one important chemical component, and which helps for the fixation of carbon dioxide in C3 cycle, ribulose 1,5 bisphosphate, RUBP. There we have seen this ribulose also. And for that there is a one enzyme is required, that is called ribulose enzyme. So there we have seen this ribulose pentose sugar, and it is a ketose form of sugar. Okay, right. So and coming to their structural diagram, if you draw means ribose sugar same as it is, but we have to draw 5 carbon like this, CHO and a C here, HOH, and here CHOH, C 1 2 3 4. So last carbon CH2. So this is the formula for ribose sugar. Now coming to ribulose, so what we have to do, just we have to add ketone group at the second carbon like this. So CH2 first carbon, that is first, and here second carbon we have ketone group, and same remaining structure is as it is like ribose, CHOH and CH2, carbon number 1, 2, 3, 4, 5, like this. And this is a structure of tribulose. And here this ribose and tribulose both are isomers to each other, okay. So, and likewise we have pentose sugar. Not only in pentose sugar aldose form, that is ribose, but also one more pentose sugar form also we have in aldose form. So that is deoxyribose. Deoxyribose. Generally, this deoxyribose sugar present in DNA, very important. Deoxyribonucleic acid, and this DNA RNA, both are nucleic acids. So in nucleic acid, generally we have seen pentose sugar. So, and remember, and this ribose and deoxyribose both are aldose form of pentose sugar. Both are aldose form of pentose sugar only. Okay, but the major differences between ribose sugar and deoxyribose sugar is, so deoxy means here at the second carbon of the ribose sugar, OH we have now like this here, but here absent of OH. For example, if you draw this structure means, for example, here I will draw here. So CHO and second carbon CHOH, but here as it is RNA, but here only oxygen is absent, only H is present, and as it is then CHOH and CHOH and C. So if you draw in a spiral ring form like this, they contain like this structures. Yeah. So carbon number 1, 2, 3, 4, 5. Okay. So at first carbon as it is here CHO means H and OH. So here O. If you add OH, then it be ribose sugar. If you remove this oxygen from the ribose sugar, only here H is present. So that H structure is nothing but deoxyribose sugar. So removal of oxygen from the OH, then it be deoxyribose. Okay. So that is the major differences between ribose sugar and deoxyribose sugar. So finally, we obtain deoxyribose sugar here like this. Simple. Like so, carbon number 1, 2, 3, 4, 5. So at second carbon, we have only hydrogen. There is no oxygen. So this is the deoxyribose sugar. So important point here, ribose sugar is present in RNA nucleic acid and deoxyribose sugar is present in DNA. And ribose and deoxyribose both are aldose form of pentose sugar. It is most important. Okay, they are pentose sugar, and very important, and deoxyribose aldose form, and which is not isomers to the any keto form. Still no, there is no discovery of ketose form of deoxyribose. So remember, no discovery of and no discovery of ketose form of deoxyribose. So remember this. And one more example we have for aldose form in pentose sugar. So here, sorry, here I am writing. So pentose sugar one. I will take another slides. Yeah. Okay. So here another example for aldose form, that is xylose. Xylose. Xylose is a aldose form of pentose sugar. Pentose sugar, and it is isomers or means ketose form of xylulose. Xylose is xylulose. So xylulose is a ketose form of pentose sugar. Pentose sugar. Remember, and in previous we discussed like ribose and deoxyribose. Deoxyribose. Both are also aldose form. Aldose form of pentose sugar. Pentose sugar. But here ribose has a ketose form, that is ribulose. But here deoxyribose doesn't have, not known. Till now there is no discovery of any ketose form of pentose sugar, that is deoxyribose sugar. Okay, remember this important part. So all these are pentose sugar. They contain 5 carbon compound. Now we move on hexose sugar. Very important, hexose sugar. Here hexose sugar. Hexose means 6 carbon. 6 carbon. If you draw the formula, like they contain 6,6H12O6. Okay. So here in hexose sugar we also have, like in hexose sugar here also we have aldose form like CHO, and also we have ketose form, the double bond. So here first we'll discuss aldose form of hexose sugar. So example, first most important example is glucose. Glucose. So glucose is the aldose form of hexose sugar. And generally, if you write the formula, same like C6H12O6. Very important. Glucose is a blood sugar. Blood sugar. Blood sugar. And for this glucose, the ketose form is fructose. Very important. Example, fructose. Fructose. So here fructose formula also as it is like C6. But what is the differences between glucose and fructose? Only because of functional group. If the functional group is aldehyde, then it become glucose. If functional group is ketone, then it become fructose. Do you know fructose is a fruit sugar? Fruit sugar. Fruit sugar. And this glucose and fructose both are isomers to each other. Means they are formed from aldol group to ketone group. And do you know, not only fruit sugar, it is also sweetest sugar. It is a sweetest sugar. And one important note here, the fructose is the only carbohydrate which synthesized in semen. It is the only carbohydrate which synthesize in semen. In semen. Generally, the semen secreted from male reproductive glands like prostate gland. So in semen only we have seen this fructose carbohydrate. It is the only carbohydrate which is synthesized in semen, that is fructose. So fructose is a ketose form of hexose sugar. So also we have various aldose form of hexose sugar, like here I am writing second one, mannose. So mannose formula also same like C6. For this mannose, the isomers is D-fructose. Very important. So D-fructose is a ketose form of mannose. And coming to third aldose form, that is galactose, like C6. So for this, isomers of our ketose form is tagatose. So tagatose is a ketose form of hexose groups. And one more and last most important fourth one, that is Ramos. Same C6 only. And for this, the isomers is ramulose. So ramulose is a ketose form. So likewise, we have different types of aldose form of hexose sugar like glucose, mannose, galactose, and ramose. And for this, we have ketose form in hexose group like fructose, defructose, tagatose, and ramulose. Okay. And very important here, generally this Ramos keto aldose form of hexose sugar, which is found in Bacillariophyceae algae, which is found in and it is found as a reserve food material in Bacillariophyceae algae. Bacillariophyceae. Generally, Bacillariophyceae are nothing but diatoms. In diatoms, the reserve food material, that is Ramos. Okay. So this is all about the hexose sugar. Now we move on another last in monosaccharide, that is heptose sugar. Heptose sugar. Here heptose means carbon number is 7. If they has carbon number 7 means if we can write the formula like C7, oxygen is 7. Heptose sugar. The best example for heptose sugar here, xylose. Sorry. Pseudoheptulose. Pseudoheptose. Pseudoheptulose. So here important note here, the pseudoheptulose is the only ketose form of heptose sugar. Ketose form of heptose sugar. It is the only ketose form of heptose sugar. It means that in heptose sugar, no aldehyde form of sugar are present, are discovered. No aldehyde form of heptose sugar are discovered. Only in heptose sugar we have seen only ketose form of heptose sugar, that is pseudo. Very important, that is pseudo. Okay. So likewise we have different types of mono. Now we move on two important concepts, like their epimers and anomers. What exactly mean of epimers and anomers are seen in monosaccharides. Here first we'll discuss about epimers. So here epimers means the carbohydrate, especially monosaccharide, which are differ at only one carbon, any one except to carbon number 1. Here I am writing the monosaccharide, mono which differ at only one carbon, which differ only at only one carbon, except, very very important, except carbon number 1, and such monosaccharides are called epimers. Here, for example, glucose hexose sugar, it has two epimers. How means, like, it has mannose, and it has galactose. Mannose and galactose both are epimers of glucose, remember. And how they are differ means, the mannose differ to the glucose at carbon number second, and this galactose differ at carbon number 4. How means, we will discuss. Generally, here I will write here. Yeah. So here I am drawing carbohydrate, glucose, CHO at second carbon, and third carbon number 1, 2, carbon number 3. Here at carbon number, in generally in glucose, carbon number 3 at left side. Here we have generally in glucose, here we have H only. Carbon number 3 only change here. And this side we have OH. At carbon number 3 only, generally glucose change here. The remaining structure is same. Like carbon number 4 has CHOH, and carbon number 5 also has CHOH, and last carbon number CH2. So this is the structure of glucose. So remember, generally glucose has some configuration, like at carbon number 3, and this side, left side we have only H, and this side we have only OH, and the remaining structure is as it is. And now this is the structure of glucose, and this glucose has two epimers. How means, if there is a change in carbon number 2, see, this is like the, here we have 6 carbon, 3, 4, 5, 6. So here, for example, I draw with another pen. Yeah. For example, at a change in carbon number 2 means here, then it become mannose. If change at carbon number 4, then it become galactose. How means, I will draw here. So here CHO, and here C, generally this side OH, no. Now here this side OH and H, sorry, H, and as it is glucose structure, C, same, CH and CH2OH. See, so there is a changes at the carbon number 2, and the remaining whole structure as it is like glucose. So if, and this is the structure of what? Mannose. If you write the galactose, how we write means like this. For example, here CHO, carbon number 1, first I will write carbon 2, carbon 3, and carbon 4, carbon number 5, and carbon number 6, right? So glucose, CHO, carbon number 2 as it is CHOH, but here carbon number 3, this side H, this side OH, usually. And here at the fourth carbon, see here, generally at fourth carbon in glucose, OH this side now, and here OH this side, and as it is H, and then CHOH and CH2OH, same. So if you look at here, and this is the epimers of glucose called galactose. It is the structure of galactose. If you observe the glucose structure at a third carbon, above the changes of third carbon, it is become mannose, and below the third carbon, if there is a change, if there is a configuration, then it become galactose. Okay, so remember, above the third carbon it become mannose, second mannose, okay, second carbon become change. But if we see the structure of glucose below the third carbon, then it become galactose. So here only we have changes. The remaining structure is as it is like glucose. So this is what we call epimers. Okay, so glucose has likewise two epimers like mannose and galactose. Okay, right. Likewise, even fructose also has some epimers. Like here, example, fructose. Here fructose also has like epimers. For example, fructose has generally 3 epimers. Very important. Fructose has 3 epimers. Here glucose has only 2 epimers, but fructose has 3 epimers. So what are the epimers of fructose means here? First, most important, that is tagatose. So here tagatose epimers are changed at carbon number 4 of fructose. And coming to second one, sarbose. So here carbon number changed at carbon number 5, and the third one is psychose. Psychose are also called psychose, also called allulose. 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 Here changes at carbon number 3. So at carbon number 4, first 3, 4, 5. If there is a changes in this configuration of fructose, and all these are the epimers of the fructose. So remember, so tagatose change at carbon number 4, sarbose at carbon number 5, and psychose at carbon number 3rd of this fructose, then it become epimers of the fructose. So this is all about the important concept. How yeah, so likewise we have epimers. Now we move on another important concept, that is anomers. Yeah, here I am writing about anomers. Sorry, yeah, anomers. So what is anomers? So anomers are, they also monosaccharides are sugar which differ at carbon number 1. Very, very important. In epimers we discuss only one carbon except to carbon number 1. But in anomers, the monosaccharide, the mono are sugar which differ at carbon number 1. Carbon number 1. And such carbon number 1 is known as a anomeric carbon. Anomeric carbon. Anomeric carbon is always carbon number 1. If there is a changes at carbon number 1 or which differ at carbon number 1, such carbohydrates are known as anomers. For example, we have like alpha glucose and beta glucose. How they are formed we will discuss. And alpha glucose and beta glucose both are anomers. Both are anomers. So in this video lecture we discuss in before isomers. So isomers, always remember, it is the interconversion between aldose form to ketose form, then it said to be as a isomers. And just now we discuss about epimers. So epimers means change at only one carbon. For example, glucose has 2 epimers, fructose has 3 epimers, like that. Now we will discuss about anomers. So anomers is change in the carbon number 1 only, and like alpha glucose and beta glucose, alpha fructose and beta fructose, like that. So how they change means we will discuss. So alpha glucose and beta glucose are the anomers of each others. For example, if we draw the glucose structure in a pyranose form, like this we have. Yeah. Yes. So here carbon number 1, carbon number 2, carbon number 3, carbon number 4, carbon number 5, carbon number 6. Hexose sugar, no glucose, right? So here generally carbon number 1 H2OH. Same like here OH H, but third carbon H2OH. This is the changes only. As it is H2H2OH, like that. It is the structure of glucose. So if you observe the structure of glucose at carbon number 1, you focus on only carbon number 1 here. Carbon number 1 here. At carbon number 1, the OH is below here. If the OH is down, and such carbohydrates are said to be as a alpha. So here alpha glucose. Alpha glucose. If you draw beta glucose, how their structures they are differ means like this. Yeah. If you draw beta glucose structure. So here also we have carbon number 1, 2, 3, and here 4 carbon, 5 carbon, and 6 carbon. Here if you interchange means at carbon number 1 only. For example, OH above and H below. Here observe this carbon number, how they are present means here like this. OH is above and H is below, like this. If you have like this structure of carbohydrate, then it said to be as a beta form. Or here we are taking as an example of glucose means, that's why here beta glucose. So if you observe the structure in alpha glucose and beta glucose, the changes are which differ only at carbon number 1. If OH is always below, then it is said to be as alpha. If it is above, then it also said to be as a beta. So okay, so this is how anomers are present. Okay, so and again, so with this we have completed carbohydrate and their uh, examples like triose sugar, tetrose sugar, pentose sugar, hexose sugar, and heptose sugar. And we have two important concepts related to the monosaccharides like epimers and anomers. And one more important concept here we need to discuss about carbohydrate, that is derivatives of carbohydrates or modification of carbohydrate, that is monosaccharide modification. So here we will discuss most important concept, that is derivatives. Derivatives of derivatives of monosaccharides are modification of mono. So in monosaccharide there are various modifications are there. So we will discuss all these derivatives of monosaccharides or modification of monosaccharide in next videos, next video lecture we'll discuss in detail of how monosaccharides get modified or and what are the derivatives of these monosaccharides. So in which we discuss mainly sugar acid, and in which we discuss how sugar acid are formed or how sugar alcohol will form and how amino sugar will form. We will discuss in depth in another video lecture. So for today, that's it for today class. We discuss each and everything concept related to the monosaccharides. Just we have left only this derivatives of monosaccharides. We will discuss in another video lecture. So here I have provided three highly important NCERT based MCQs, multi-statement MCQs here. Yeah. So read these MCQs one. Yeah. So consider the following statement regarding the glucose and fructose. We are asking about glucose and fructose. Remember, glucose and fructose both are hexose sugar, and both are what? Glucose and fructose both are aldose form or ketose form. So D-glucose, D-fructose. Likewise, here there are four options are there. Read these four options and choose the correct options. Yeah. Coming to another MCQs here I have provided to you. Yeah. Consider the following statement regarding the monosaccharide derivatives. Actually, we not discuss, but in another video lecture, we will discuss derivatives of monosaccharides. Oh, sorry. Here, last MCQs from this topic. So, pair of cyclics stereoisomers of D-glucose that differ in configuration only at carbon number 1. Here, differ only at carbon number 1. So, highlight this carbon number 1 atom only, which is differ. Highlight this point. So, what we call epimers, enantiomers, anomers are geometrical isomers. So, that's it. So if you like my videos, so please subscribe my channel and please share these videos to your friends. So thank you. Have a nice day.